Genetic engineering researchers have discovered a very powerful tool that can be used to edit genes on a much larger scale than CRISPR, allowing for the ability to rearrange, recombine, invert, duplicate, move, and perform other editing operations on large DNA sequences. This is a next generation genomic design method and goes much beyond what CRISPR and other genetic editing tools can do. It is called the bridge recombinase mechanism, or the RNA bridge, and it is a completely novel method that in fact utilizes what exists naturally in our genetic machinery, a bit of DNA that can program itself to edit other large DNA sequences. It utilizes mobile genetic elements or jumping genes which cut and paste themselves into genomes all over our bodies and are present in all forms of life, and they perform on-the-go DNA manipulation in all living beings every day. The findings are reported in the form of two papers characterizing the discovery. One talks about the working of the RNA bridge, the discovery, the molecule itself, and how it can be reprogrammed as needed. And the other paper talks about the structural mechanism behind the discovered recombination ability of these genes and the steps that they break down into. Jumping genes are minimal segments of DNA that have the recombinase enzyme, which helps bind this bit of jumping gene DNA to other DNA, along with extra DNA segments at the end. The team discovered that this bit of flanking extra DNA at the ends of the jumping genes in fact get joined together when the gene is not in action. It then converts the double helical DNA structure of this jumping gene into a single strand RNA molecule. This molecule then folds into two loops. These two loops can then independently bind to a donor DNA and a target DNA. These two loops can bind independently to a donor DNA segment and a target DNA segment. Thus, the jumping gene actually functions as a physical bridge that recombines two bits of unconnected DNA. Once this bridging is done, this jumping gene can then cut itself out of this DNA segment and move around the rest of the body while the two bits of DNA remain connected. The researchers also made the important discovery that the donor loop and the target loop can be programmed independently. This offers great flexibility in inserting or recombining sequences of DNA. The name of the jumping gene is IS110, which stands for insertion sequence 110. Such insertion sequences are found in ample quantities in bacteria, which is what was used for the study, specifically the E. coli bacteria. These insertion sequences or jumping genes roam around the body, cutting and pasting themselves, repairing DNA and modifying it on a daily basis. IS110 bridge recombination system expands the ability of nucleic acid guided systems, that is, systems that work on RNA and DNA. The bridge method is greatly flexible to perform specifically three fundamental DNA editing operations, which are insertion, inversion, and excision or removal. The role of the flanking DNA, the extra bits of DNA present at the end of these jumping genes around the recombinase genes of these mobile sequences, has been understood properly for the first time in this study. One of the authors of the paper compared the bridge RNA to a universal power adapter that makes the IS110 compatible to any outlet, or in this case, a DNA segment. The second paper reports results from studying this mechanism through cryo-electron microscopy, and it shows that the recombination occurs in three different steps, and we're not really going into that detail in this video. So how does this system compare today? Both existing CRISPR and RNAi or RNA interface, which is another form of gene editing, work by blocking or activating gene expression. That is, they go and modify genes to either get them to do a function or to prevent them from doing what they're doing. RNA interface does this by targeting RNA molecules, which work like instruction sets, which then go and do the work of editing the DNA. The CRISPR method directly targets DNA itself instead of involving RNA in between. Now, this RNA bridge, on the other hand, modifies large sequences of DNA instead of just a tiny segment consisting of a handful of genes like RNA interface and CRISPR do. Therefore, this greatly expands the scope of changes that can be wielded by the technique, which is also a limitation of the method itself. Since the technique modifies large chunks of DNA sequences, there is a high risk of unintentional consequences. This disadvantage doesn't present itself to this scale with CRISPR. Another limitation of the findings is that they were performed in vitro or in the lab on bacteria. To apply the RNA bridge to any humans and modify human sequences, the method would need to be tried in animal models and mammal models first. And that is where the research is going. While this is a path-breaking discovery for our gene editing techniques, the next 5 to 10 years will see more research and more bits of news breaking about the developments that have been done with the RNA bridge mechanism before they can be used in humans for therapeutics and treatment.